We all are familiar with the term of microbes. So let's enhance our knowledge regarding the terms of microbes in human welfare. What is this? How can this virus came here? This stupid virus? This dangerous and killer virus? How can this came here? Where's my mask? Where's the hand sanitizer? Oh, alas, this virus is gone. ₹50 cut over acting. So finally, it's time for going with human welfare. So, here's term microbes refers to those organisms which generally we can't view with our naked eyes. It is only visible under the microscope. Maybe it is a compound microscope, maybe it's an electronic microscope, or maybe it's a simple microscope too. Microbes in this diversified world act as both friends and enemies. Now today in this module, we are going to deal with the cases where microbes are referred to as our friends. In this entire huge world, there are various cases, but we are going to deal with some few cases only. So, the first topic goes like this, it is lactic acid bacteria. It is referred to as lactic acid bacteria, in short, LAB. This LAB is basically used for curd production. We all know from our junior classes also. But what is the main phenomenon here? The main funda here is something like this. What happens in this case, when we take some curd as inoculum and put it in a fresh purified milk, what happens is that the casein protein present in the milk gets coagulated and there forms three layers in the solution jar. The first top layer is of the butter, the second is of the water, and the third layer is of the lassi. These three mixtures, when are coagulated and begins in a condensed solidified form, it is referred to as curd, which we take in our day-to-day life. The next point goes with the Saccharomyces cerevisiae. Saccharomyces cerevisiae is also termed as brewers yeast. You may check the photo here. It is usually used for bread making. What is the main implementation of it? Is that the wheat or flour is taken and some amount of yeast is generally available in our environment, okay, in the atmosphere only. So no need to put some additional yeast in a general life, but for commercial purposes, yeast powder added to it. Yeast present in the yeast powder when it reacts with the entire material, the system. What happens is that the carbon present in the material substance gets reacted with some oxygen molecule in the environment and comes out of the substance. So due to this coming out of the substance, there is a fluffiness or dough-like substance creating in the entire mixture, due to which it gets fluffed and we get a fermented material from which we prepare our day-to-day lives in our various tasty, delicious foods. Now, bread making is also done with the same process also. Now, the third one is our Propionibacteria sermani. This Propionibacteria sermani is referred to as a very useful bacterium, as it is a very costly bacterium also. As we know that cheese is a very costly substance. At least there is a fact that approximately 10 liters of milk are used to produce 1 liter of cheese. So it is very costly also. Now, Swiss cheese is basically one delicious and more tasty sweet. Yes, it is a very tasty cheese only. So what happens in that cheese is that due to the presence of this Propionibacteria sermani, some gases are formed in the system which tries to come out and from the surface area of the cheese, and due to which some holes or hollow surfaces are created in the cheese substance. Due to which, further we see that when we get the cheese is basically a mixture of huge concentration of fatty acids and some nutrients, as majority of the proteins and all have already gone away from the cheese. Mainly it consists of fatty acids and some nutrients also. So it is very tasty. Fourth factor goes with the Penicillium notatum. Now this is quite interesting and important. Penicillium is notatum. Penicillium notatum is a fungus. Okay. Now it is used to produce penicillin, which we are dealing with from our GK classes in our class 5-6. But then do we know it? So let's get into the depth of it. That Penicillium notatum is something which acts as an antibacterial drug. This antibacterial drug was first invented very years ago, and it was the first discovered antibacterial dose, or normally to say antibiotic. This Penicillium notatum basically acts in staphylococcus. It was observed by the scientist that when this Penicillium notatum is kept in a medium containing staphylococcus, the staphylococcus are resistant to it and going away it or either die or going away of it. So what was seen that due to this Penicillium notatum, the staphylococcus was dying. So this experiment was continued further in human blood system, and which was seen that it is Penicillium notatum. Humans are also being helpful to prevent the staphylococcus attacks. So we see the structure of Penicillium notatum in a chemical formula, something like this, in this image. Now the next one goes with the Aspergillus niger. Aspergillus niger is also a very important term. Aspergillus niger is a microbe which is generally used for the production of citric acid. Now citric acid is something very much essential for us, but it may be manufactured too with the help of Aspergillus niger. But it is generally not done so much as citric acid is generally present in our day-to-day life in a huge quantity. But then too, the biotechnicians in some perfect implementation, they use this. The next factor goes with the Acidobacter acidae. Acidobacter acidae is the basic microbe which is used for the production of acetic acid. This is very much important acid and very much interesting and very much useful for the mankind too. The next goes with the Clostridium butylicum. As we can go with the name, just try to focus on the name, that is Clostridium butylicum. Butylicum is quite similar with butyric acid, and this Acidobacter acid is similar with acetic acid. So you can quite remember easily that acetic acid means acetic acid, butyric acid. Important factors, some important terms is Streptococcus, Trichoderma polysporum, and Monasca purpurus. These three microbes are the most important microbes in the entire chapter. Quite to dealing with them, that these three microbes are the main contributing factor of the questions of every exam, being it our board examination, being it our comparative examinations. If we are going with the past year papers, past year question papers, PIQs, we will surely getting that these names are very much, questions are very much common from this three terms, Streptococcus, Trichoderma polysporum, Monasca purpurus, and these are very much important to in day-to-day life. So the first one is being the Streptococcus is very important, as Streptococcus is microbe producing streptokinase, that is streptokinase. Streptococcus produces streptokinase. On R is here, streptokinase. Streptokinase is having a very important medical application. The medical application is something that it helps in blood clot busting. From the time it has been discovered, it has been being used for the accidental trauma cases. Okay, at this time we are having various advancement in our technology, so we are getting some other enzymes, other chemicals also. But this was the initial stage. Trichoderma polysporum. This Trichoderma polysporum is also very important, as it is being a fungus, helps in production of cyclosporin A. Cyclosporin A is very much important, as it is basically used as immunosuppression. I am getting the idea, I am giving a bit idea, example of it, that when at the time of organ transplantation, the patient is supplied or operated with an organ from any other donor, okay, at that time the body of the patient starts producing antibodies against the foreign organ which have been donated by the other donor, as it is not a part of the same body, human being also, as it is not a part of the same human being. So at that time, if this activeness of the inhibitors or the antibodies gets to a huge extent, then the organ which is being operated from the donor and supplied to the patient will be damaged and may be destroyed. So the entire operation will get into a failure rather than being a success after being so much hectic being of operation. So to prevent that case, the doctors used to give immunosuppressions. At the time was there when this Trichoderma polysporum was used as immunosuppression. Till date it is being used, but there are various advancements in our technology, in our medical science, in our biotechnology field. So some other factors are also used, but it is a very prominent factor as immunosuppression. Now the next one goes with the Monascus purpurus. It is very, very important. Monascus purpurus produces statins. These statins are basically used as fat or cholesterol burning. Okay, it is blood cholesterol lowering agent. The arteries and the arteries get some cholesterol decompositions which gets in the blood, which create problems for the heart to pump the blood in a correct manner. So to prepare it, it may be obvious to be operated. It is sometimes it is also to be operated, but generally, in some cases, to prevent any severe heart attack, the medicines which are given to the patient may include this monocyclus bacillus, as this contains statins which can act as a blood cholesterol lowering agent. It will reduce the cholesterol amount in the blood. It will surely enhance no further decomposition of blood cholesterol in a particular junction and creating huge pressure for the blood to burst the arteriole and create a problem for the heart. So this changes. Okay. There is trichoderma species. Other than polysporum, trichoderma is having other species also. It is similar to other organisms, other microbes also. Any microbes is having made various species. Okay. So polysporum acts as something as for cyclosporin A, but trichoderma species, other species or fungus species, acts are having various huge applications. Some of them acts as effective biocontrol agent for plant pathogens. Trichoderma species, this fungus species, is also effective for biocontrol agent for giving natural pesticide other than this chemical pesticides. So efficient and non-destructive, non-toxic for mankind. And it is being a pesticide, it is non-toxic to mankind. Now this last segment consists of some important microbes, name of some important microbes with their function, in which one is our Bacillus thuringiensis. Bacillus thuringiensis is having its detailed analysis in the chapter of application of biotechnology. But to explain to you to a bit extent, that is something looking like as a insecticide, as this Bacillus thuringiensis is having the capability of killing the insects, basically the caterpillars, okay, caterpillars and beetles and all those, as it is having a cry gene, cry gene. This cry gene is basically present in the bacteria. So what does the scientist did? To kill the pests acting on the plants, they developed a new idea using biotechnology, that this cry gene was extracted from the bacteria and was put into the gene of the plant cell. Now what was seen, that the plant cell was developed with some new factors by having more capability to resist the attack of the foreign organisms, which included cotton bollworms, caterpillars, moths, etc. And they basically did what is that enhanced the production and made reduction in the post-larval losses. The next one goes with the nucleopolyhedrovirus. Another species related with them. This nucleopolyhedrovirus is a very interesting virus. Okay, this nucleopolyhedrovirus also referred as baculoviruses. They are basically having some species-specific narrow spectrum interaction. I will give you an example. We all know dragonflies. Now dragonflies are basically used in normal cases in the farms as they feed on mosquitoes. Mosquitoes, malarial mosquitoes, dengue mosquitoes. This dragonflies may also be affected with some insecticides, as this insecticide, other insecticides not having so species specific interaction, so they may kill the dragonfly also. But this baculovirus having the capability of some species specific interaction. They will be only feeding on some particular pest, not on the dragonflies or the red dotted beetles and other organisms too. So they will be specifically saving the nature and directly or indirectly the mankind. So this is very interesting and important. It is included in the modern biotechnological application in the modern pharmacological actions also. Next one goes to the rhizobium. This rhizobium, azospirillum, azorobacter and cyanobacteria. These three categories are basically conclusive for nitrogen fixation. They may fix atmospheric nitrogen or soil nitrogen, present in water and everything. It will basically related to nitrogen fixation. They are having huge number of similarities. But I mean minorities too, dissimilarities too. These dissimilarities are resultant due to the atmosphere in which they survive. As because some survive in the free atmosphere, some in the soil, some in water or free living states, anything. Apart from all those, they are having a common function of nitrogen fixation. They fix atmospheric nitrogen, convert them into organic modules, and organic modules add fertility to the soil and finally enhance the fertility to the soil to give the plant present in that soil a big growth and a huge production. So these are very important. The cyanobacteria consists of Anabaena nostomoxidria, which we know from our basic class that these three are having the capacity of nitrogen fixation. They are basically found in the roots of the leguminous plants, the protein plant, there's dal plants and everything, all those, the pea plants. Now, last but not the least is having the symbiosis. Let's deal with the mycorrhiza. So what is mycorrhiza? Mycorrhiza is a very much important symbiotic relationship between fungus and plants. So what happens is in this relationship, the fungus helps in absorption of the phosphorus present in the soil and transmits it to the roots of the plant. The fungus exists in the roots of the plant only. So the plants getting that phosphorus extracted by the fungus transmits it to the photosynthetic cycle, and what happens is that it gets an enhancement. As we know that phosphorus enhances the production of food and the growth development, so it enhances the growth development also. As a gift to the fungus, the plant supplies nutrients to it. So in this regard, the fungus and plant gets symbiosis and forms the system of mycorrhiza. You can get the images of mycorrhiza like over here, just over here. This entire chart is completed. But in this entire chapter, two more topics are left. The first one is our biogas production, and second one is our sewage treatment. This is municipal sewage treatment. So for biogas production, we know of the huge biogas bioreactors, biogas plants. What happens in the villages? It was first produced by this huge biogas plants. What happens is that the excessive cow dung and slurp, rotten materials of the domestic household are being deposited in a big chamber, which acts as a slur over there. Due to this and the anaerobic condition, huge temperature inside it, the methanogen or anaerobic bacteria act on the slur, and due to the action of them on the slur, what happens is that huge amount of methane, H2S, and a bit carbon dioxide is produced. 70 to 80% is methane in the mixture, and H2S is approximately 13%. So this huge, highly flammable mixture acts as a huge source of energy for the domestic household in the villages. It has been working since many days, and till date it has been working in some of the villages of our country. It's a huge natural source of energy. The next treatment is of the sewage treatment. This something looks like this. This sewage chamber is very interesting. There have been three steps. Primary step, secondary step, and tertiary step. Primary, secondary, and tertiary are having various processes. It happens in the huge municipal corporations that the drainage and the sewage which is coming out of every household is being treated in the sewage plants since many days. The primary treatment, that huge sewage is being taken into huge chambers over which primary sedimentation occurs. Means the huge particles present, the heavy particles, get sedimented and forms a slur. After overflowing it is being slowly transferred to another chamber and it is being treated to further treatment or the filtration treatment, which we have dealt in 9 and 10 and junior classes also. So now the main concern of today over here is with this primary slur which is being deposited in the material. So what happens is that in the slur is being taken to the secondary chamber. In the secondary chamber, the aerobic bacteria are present, which totally digest the organic matter present in the soil. The organic matter which is being present in the slur is being digested gradually, due to which some amounts of methane, H2S, carbon dioxide and similar gases are evolved. Now the partially filtered slur is again being put to the tertiary chamber. In the tertiary chamber, anaerobic bacteria are present, like methanogens. This anaerobic bacteria, what does is that they digest the remaining organic matter and also the aerobic bacteria, which has done the previous graduation. So by this, it nearly gets filtrated to 70% extent. There is huge extent. So after that time, it is a perfect biodegradable mixture. So it can be deposited or decomposed or disposed into any natural body. What happens in the normal cases is that the mixture is being transmitted to the huge water flings, to the huge water bodies like the rivers, the lakes, and all those. In them, they are safely deposited, and the Mother Earth can take them as they are biodegradable at that time. Now, what happens in our day-to-day life and this present scenario, what is happening in the present world is that the population is increasing to a bit extent, but the number of sewage plants is being constant. They are not increased by the government or other sectors. So this small amount of sewage treatments plants are having so much pressure to treat the huge amount of sewage coming from this entire population. So due to that great pressure, it is very obvious that majority of the sewage is being directly disposed to the water bodies without being filtered, which creates a great pollution and massacres in the environment. It is a prime and it is a point of great concern for our government to take this. So let's see what happens in the coming future. With this chapter has been completed total for our board examinations as well as our NEET examination too. I will be sharing this entire chart as the PDF in the first comment. So what you have to do is that share this channel and hit that subscribe button below. The red button below. Please subscribe, click it, and don't forget to press the bell icon. Just press it beside it and share it as much as you can. So keep studying.